Welcome to my symposium piece. My name is Parker, and today I'll be talking about the work I've done this year in the Steinel lab. The work I've been doing has been on tardigrades, their immune responses, and their parasites. At the start, I began looking into identifying tardigrades and the parasites that can be found on them, so it could be seen if an immune response occurred in response to being parasitized. Identification of tardigrades generally begins with observing the buccal apparatus, which is the tube the tardigrade eats through, cephalic appendages, which look like tentacles on their backs, and the feet of the tardigrade, which can have suckers or claws. For determining the difference between different species, DNA analysis is often used. All tardigrades displayed in these slides are in the class Tardigrada, as they reside in a semi-aquatic environment, have no cephalic appendages, and have claws. The tardigrade in this slide is likely in the order Parachila, as has a slightly bent portion to the buccal apparatus that has some spiraling. I'm still working on what genus this particular tardigrade is from, as its claws haven't yet matched any of the genus of Parachila I've looked at so far. On the topic of looking for parasites on the tardigrades, I did not cite any fungus or other organisms on the tardigrades I've imaged. Moving forward, I've begun working on a new question. Do tardigrades produce reactive oxygen species during immune response? In most animals, ROS is utilized by the immune system for cell signaling and killing pathogens. In terms of signaling, ROS is often used for propagating inflammation. ROS can also be used to kill pathogens by inflicting oxidative damage on all pathogens they are generated near. To observe the level of ROS present in each tardigrade, I plan to use the fluorescent indicator DHR123, which fluoresces in the presence of ROS. In order to mimic an infection, LPS, which is a piece of a bacterial cell wall, will be introduced to some of the tardigrades as well. If tardigrades produce reactive oxygen species to assist in fighting off infections, then an increase in ROS should be visible through greater fluorescence in the tardigrades that are exposed to LPS. Those that are not given LPS should provide a benchmark to compare the fluorescence levels of the LPS-exposed tardigrades to. Overall, I think it is extremely likely that the tardigrades will produce ROS in response to LPS, as ROS is generally involved in the innate immune system, which all animals have to some extent. That's all I have for now. Thank you for letting me share what I've been working on with you. Have a great day.